Let's move to our next speaker, who represents the Institute of Molecular Medicine in Finland. Welcome. And let's see the video of the Institute first.
Why do some of us fall ill while others stay healthy? And why does a treatment cure one patient but not another? At the Institute for Molecular Medicine Finland, we dive deep into these questions every day. Our mission is to improve individual health outcomes through groundbreaking research in disease mechanisms to predict, prevent, and treat illness. Our journey is powered by a diverse team of experts from medicine, data science, biosciences, and beyond. Together, we harness our multidisciplinary strengths to unlock the complexities of human health. At FIMM, we utilize unique population-scale finished genomic and health data sources and cutting-edge technology, striving to translate our scientific findings into clinical practice and new therapies that benefit individuals across the globe. Our passion extends beyond research. We are educators, nurturing the next wave of scientists and advocates, actively engaging the wider community. We believe in the power of team science to bring the promise of precision health to life.
Rhythms shapes us. From sleep cycles to gene expression, Lea Urpa draws connections between music, biology, and time, inviting us to hear the hidden rhythms of our genomes. Please welcome here Lea. And let's try to connect with her colleagues in Helsinki. Hello, guys. Thank you for joining us. Thank you for your support for Lea, and thanks for following the whole contest online. Lea, the floor is yours.
Musica universalis is an ancient philosophical concept that claimed that the movement of celestial bodies followed precise mathematical equations, producing an inaudible harmony of music in the heavens. While it turns out there are few celestial bodies in harmonic resonance in our solar system, many exist in the universe, like this planetary system, over 200 light years away, depicted here in an artist's rendition, giving each resonance a sound on a melodic scale. The term harmonic originates in music theory, where a plucked string vibrates not just at the wavelength of the string itself, here at 100 hertz, but at integer multiplications of the so-called fundamental frequency, such as 200, 400, and 800 hertz. Summing these harmonics generates a composite waveform that looks distorted, but is actually much closer to our perception of music than the individual tones. This is because pure oscillation with a single frequency rarely exists in nature. Physical rhythms are often the compilation of many superimposed harmonic oscillations, such as the harmonic resonances found in music, light, and the laws of planetary motion. And biology is no exception. The best-known rhythm to biology and physiology is one that's intimately familiar to every one of us here, the 24-hour circadian rhythm. Within each of our bodies is an intrinsic circadian clock that orchestrates physiological processes that adapt the internal environment with external cues, with a well-characterized molecular clock. But there's also tantalizing evidence of so-called ultraradian rhythms, oscillations at shorter harmonics of 24 hours, as seen here in the oscillating transcription of genes in the mouse liver at 12 and 8, in addition to 24 hours. These studies rely on the sampling of tissues at frequent time points in animals, which makes the study of rhythmic biological oscillations in humans a little bit challenging. But the last decade's improvement in omics technologies has made assessing biology at the population scale possible, such as in the FINGEN project's analysis of 10% of the human population. These data have been invaluable in finding, in particular, connections between genetic variation, the differences in DNA code in individuals that set before we're born, and different diseases and other traits. Yet these studies usually ignore time of day and other cyclical oscillations in biological variation. Normally in a genome-wide association study, we make an association with a genetic variant, for example here on chromosome 11, where about half of individuals have a G instead of a C at this position. When we test for the association between having a G instead of a C with blood glucose levels, which could be positive, increasing blood glucose levels, or negative, decreasing blood glucose levels, we don't see a significant effect either way. But when we add the dimension of time into our analysis, we see that the folks who have a G instead of a C at this position on chromosome 11 have a positive association, or increased blood glucose levels in the morning, but negative association, or decreased blood glucose levels in the evening. This shows that the previously ignored dimension of time intersects with known biology to explain just a bit more of the incredible variance in real human populations. However, this analysis is still rather reductionist, and further analysis could include other omics data like proteomics or metabolomics data, and take longitudinal electronic health records into account. Overall, our goal is to understand the biological mechanisms in the health of disease by looking at data holistically. Imagine the human body as an orchestra. Each instrument is a biological system that plays its part, and the players are the genetic programs that pluck the strings of the system. Traditional experiments pluck one string at a time, studying or manipulating a single string or a single instrument. What we aim to do is to understand the system by listening to the whole orchestra, the interplay between instruments and how it changes over time to better understand the entire system. We now have the data to do this, and only by listening to the full symphony of biology, across systems, across time, can we truly understand the music of life.
That was Lea with her presentation about rhythms of life.